Good afternoon and welcome to the very first session of CMN Live. My name is Karen O'Hanlon-Court and I am joined today by our special guest, Dr. Eric Kmitsch. Eric is the Scientific Director at the Gene Editing Institute at Christiana Care in Delaware, where he has been since 2015, following a very rich and exciting industrial and academic career at various institutes across the United States. Besides his current role at Christiana Care, Eric is also the Chief Scientific Officer at Therapeutics, which is developing novel therapies for cancer based on in vivo gene editing. So good afternoon or good morning to you, Eric. I'm absolutely delighted to have you with us today in the virtual studio.
Thanks, Karen. It's absolutely great to be with you, and good afternoon and good morning to wherever everybody is.
So today we're going to talk about therapeutic gene editing, a lot done and a lot more to do. So there's also quite a bit to talk about in that. So if it's okay with you, we'll just dive straight in.
Absolutely.
Great. So let's start first with what has been done. So we've seen the first regulatory approval for gene editing therapy with the approval of Casgevy to Vertex Pharmaceuticals and CRISPR Therapeutics just before Christmas. As somebody who has worked in the gene editing field for 25 years or so, what does this massive milestone mean to you?
Well, I think speaking for everyone who has been a part of this journey, it is totally gratifying from the beginning, sort of seeing some very basic mechanistic work evolve and watching decades of interesting work at the bench side. It's wonderful to see this come to fruition, and obviously we're cheering this kind of development and future developments along quite strongly.
Yeah, I think you're right. I think it's been amazing to see the first approval. And actually, on that note, I think it's fair to say there's been a lot of hype in the last years about how fast the field has moved, the gene editing field, since the first demonstration of CRISPR gene editing in eukaryotic cells some 12 years ago now. We've seen the Nobel Prize being awarded for CRISPR, and we have well more than 150 ongoing clinical trials spanning not just CRISPR, but also base editing and other emerging modalities. But in your opinion, has it really gone that fast? I mean, we've seen the first approval now, but gene editing has actually been around for quite some time. And if I'm not wrong, sickle cell disease was probably among the original targets.
Well, I'm living proof that it hasn't gone that fast. The gray hair I have has come from a lot of years in this field. I think it started off in model organisms, lower eukaryotes, where people were able to generate genetic knockouts using viral vectors and using DNA sequences. And I think the hope was always that we would be able to achieve the efficiency that was needed to actually repair genes. Through the years, that became much more challenging, both because the repair was not exactly accurate, but with the evolution of programmable nucleases, really starting with meganucleases in yeast to zinc finger nucleases and the pioneering work done by people in that zone of the field, and then obviously talons. And then when CRISPR came on board, that became a very, it is a tool that we all started to believe could improve the precision and efficiency. Sickle cell, I think, has always been the disease that people wanted to go after. So decades of work in trying to repair it, and then obviously Stuart Orkin's work, understanding the globin locus and the fact that there was a repressor of fetal globin expression, really led to the evolution of what we see today and the approved therapy. So I think this is true for just about every form of major breakthrough in any field. There's decades of stuff behind it, and sickle cell had always been sort of an excellent disease target.
Yeah, so it takes time. I should also have mentioned that there are, of course, ongoing trials involving talons, meganucleases, and others. It wasn't to leave those out there. You mentioned the work of Stuart Orkin, and obviously that has been instrumental in paving the way for the approaches that we've seen to curing or treating at least sickle cell disease and also thalassemia. So just to go a little bit deeper into Casgevy, so many of our people watching in will know that the therapeutic strategy underlying Casgevy doesn't involve correcting the disease-causing mutations. For anyone who's not familiar with sickle cell disease and beta thalassemia, these diseases are caused collectively by mutations that either lead to defective adult hemoglobin, which causes the cells to sickle and clump together, or an absence, or should I say, a deficiency of functional adult hemoglobin. Where Casgevy comes in, it reactivates the expression of another form of of hemoglobin, so a fetal hemoglobin, which is normally repressed earlier in life, and that's some of the repression that you were referring to. And Casgevy reactivates the fetal hemoglobin, which then compensates for the lack of functional adult hemoglobin. We've seen that from the clinical trials of Casgevy. So what kind of other target would this approach be useful for, in your opinion? And I suppose there may be people out there wondering, now we have CRISPR, we have base editing, why not just go in and actually correct the mutation or replace the gene if there are several mutations involved?
Yeah, let me take that question in sort of two pieces. The correct, obviously those of us in this field and historically those of us who labored in this field for decades always want to get to the absolute gene editing, the genetic spell checking effect, which is fixing a letter in a word or a single nucleotide in a gene analogous to the word. And our lab and others, many others, sort of found that to be really challenging. And we couldn't quite even get to the right vector, including combining, CRISPR and oligonucleotides. I think there's some hope now with prime editing or base editing going forward, because it doesn't necessarily involve the breakage of the DNA. Fundamentally, CRISPR is a remarkably precise DNA breaker. It cuts DNA and then the cell does the rest. So from the standpoint of correcting a gene, there are two major steps there: breaking, isolation, and then obviously repairing the genetic code or spell checking the word and correcting it. That's challenging. Multiple reagents are involved, the CRISPR to desiccate the site, and then the repair template, whether that comes from an adjacent chromosome or from an exogenously added oligonucleotide. I think there's incredible excitement around the natural use of CRISPR. So CRISPR emanates from prokaryotes, and there's an enormous amount of search everywhere, even the bottom of the ocean, bottom of gold mines, even in the tundra of Ireland there for new bacterial strains that contain new Cas proteins, but it naturally breaks DNA. So there are a series of diseases. We work on an approach in oncology where disabling genes can then enhance the activity of standard of care therapies much better, because disease resistance, drug resistance, these kinds of things are generally controlled, at least at some level, at the genetic level. So it's sort of the combination of using what CRISPR does naturally, which is disabled DNA, and in our case, as it gets democratized into mammalian cells, the possibility is that it now can disable genes that are preventing good therapy from working or enhancing a fact that would be beneficial to the mammalian cells. So yes, I think there are a lot of opportunities and a lot of good targets following in kind of the Vertex CRISPR Therapeutics modality of disabling a repression activity, in this case, a repressor of fetal globin expression.
Yeah. It's really interesting to hear you mentioning cancer in this context, because I think a lot of the focus has been on, CRISPR can cure rare diseases that are currently completely untreatable, and, diseases of childhood and things where they're really until now. And also with gene therapy as well, has also given fresh hopes to those kind of diseases. And I think we're still in the very early days of gene editing therapies, even though there are many trials ongoing. We've seen the first approvals. So I wonder, given your experience and your expertise in this area, where do you see that therapeutic gene editing is likely to make the biggest impact?
I think it gives hope for people who had no hope. I think there are inherited diseases that are just, pharmaceuticals have failed largely. Even before the approval of the sickle cell approach here, many sickle cell patients were on hydroxyurea. That was the prescribed treatment, which is horrible. And so I think that's the main hope. And what I've always believed, regardless of of the financial or the scientific challenges and burdens that we face in developing these new type of drugs is patient suffering counts. And
Mm.
so if you can relieve that, whether it's expensive or not, I think as humankind, we have a responsibility to develop drugs to reduce that. So I think genetic medicine, whether it's gene editing, whether it's gene therapy using adeno-associated viruses or any sort of gene augmentation, the goal here is to tackle, diseases or uh, maladies that are essentially not well handled by pharmaceuticals. Um, and of course, you mentioned oncology. It's sort of where we thought that, some of this would go, that, patients who receive chemotherapy or radiation or immunotherapy or whatever, eventually side effects occur. Or as we were talking a couple days ago, the preparation even for the sickle cell approach is significant, and these patients have to sort of wear down this process of getting ready for these standard approaches, whether it's immunosuppression. So I think gene editing gives hope to those where diseases have not been hopeful. There have been not many paths, but also has an opportunity to break into some new approaches in cancer care that eventually have seemed to be impossible to approach.
Yeah, I agree. I think there's certainly lots to take hold of, and we are still in the very early days, and we really have no idea how far and broad this can go. Also to the chronic diseases that people seem to be living with or accepting a certain level of quality of life with, but can hopefully be much better for those. I wanted to touch on something about the hydroxyurea and the current standard of care for sickle cell. There have been a few new approvals for sickle cell in the last few years, different, monoclonal antibodies to take down some of the inflammatory processes. There is also something that increases the affinity of the hemoglobin to the oxygen, and then there's an L-glutamine-based medicine
Right.
that's supposed to combat the oxidative stress. But all of those therapies still require frequent administration. Some of them are daily or injectables, which puts a burden on the patients to also show up at the clinic. So I suppose that's one aspect of why a one and done approach is so much more attractive. Nevertheless, those one and done approaches are very expensive, which you also sort of hinted at there. I've been reading a few articles about the high cost of Casgevy, but also Lovocell that was approved to Bluebird Bio. So a gene therapy for anyone who's not familiar with it, which is essentially ultimately resulting in the same outcome as Casgevy for sickle cell patients, but where they're receiving a functional copy of the globin gene on a viral vector. Both of those therapies are coming in at over $2 million. And there has been, concern amongst in the field about whether those treatments would even get to the people who need them the most. So how do you think we can solve the cost issue? Can we at all? And is it eventually going to be detrimental to the field of genetic medicine?
There's no doubt there's sticker shock when we reduce this care to patients. I think our hope is that eventually, as in the case of immunotherapy and T cell therapy, cell therapy, that the cost will eventually come down as people are able to manufacture these reagents better. It's pretty expensive to make biologics still, and
Mm.
there aren't a lot of really reliable sources of manufacturing. So there is an emphasis, certainly in Ireland, which is quite famous for their biologics manufacturing and pharmaceutical industry, but also in the United States to improve biomanufacturing. The universities are taking a very serious dive into this. So I think our hope is that at some point the manufacturing costs will be reduced. But again, I just want to circle back to this something that I learned. So Christiana Care has this really their medical approach overall is patient first strategies, and we've kind of adopted that. And so it's really hard to put a cost on patient suffering, and by reducing that, that's important. And that all sounds great, but I get your point about the practical side. Who's really going to pay for it? So I'm going to have to leave it to my glass half full optimism
Yeah.
that at some point the manufacturing costs of these gene editing entities will be reduced. No doubt, sickle cell, which is a global disease, was an interesting choice to go first because, it's still a rampant problem in Africa. And, applications are not like we have in Ireland, Britain, or the United States. So again, that transition to where it will affect people, in Africa and globally will be interesting to watch.
Yeah, I think it's also important to put into context that the average cost of treating a patient over the
Yep.
course of their lifetime with sickle cell or beta thalassemia is estimated to be between $4 and $6 million,
Yep.
plus a lot of additional costs in getting to the hospital and maybe overnight stays in hotels and all the other things that come with having chronic illness that's been probably at best poorly managed. So I think that's also important to take into account. And I suppose also maybe the autologous versus the allogeneic, that whole thing is going to maybe play out also in favor of cost. Do you think that we're going
Yeah,
to?
I do. Well, we're certainly seeing the race to develop the universal T cell, right? Cell therapy is very expensive. It works, and sometimes it doesn't work. But if you could have an off-the-shelf T cell or an off-the-shelf, progenitor cell for hemoglobin, that would be enormously valuable. So there's a whole area of research that's currently being developed that I think tangentially will enter the fray, and by its very nature and by its very product will definitely reduce costs. And that's a very good point. I think we tend to look, in my world, we look at cancer care and the enormous number of suffering, but cost that incurs somebody who has a lingering chronic cancer for 10 years. And if that can be shortened and cost reduced using a form of gene editing, that would definitely be beneficial financially, as well as improving the quality of life of the patient.
Yeah, and I think it's also just to be fair, it's not just gene editing that suffers from the high cost. It's the same with gene therapy.
Absolutely.
And part of the problem is that the diseases are so rare and there's so few that could potentially be treated. So I also hope that the two fields can learn from each other. They're not that different after all, really. So there will probably be some shared lessons there. It feels like we're moving into the a lot more to do part of the discussion.
All right. Yeah.
So, I mean, there's probably lots to unpack here. We can't get through it all today, but there's one particular thing I'd like to talk about, and that is something that comes up again and again when we interview scientists from academia, but also companies. That is the delivery issue. So I think we've seen a lot of progress in recent years. We now have viral and emerging non-viral approaches to choose from, some of which are already in the clinic, like we have the nanoparticles, the lipid nanoparticles. But it still seems like delivery is still remaining to be a major bottleneck in the field, especially for those in vivo therapies that need to go somewhere else beyond the liver. This may be even more so the case for the really difficult to target organs like the brain, which is enclosed by the very protective blood-brain barrier. So given that the toolbox of delivery methods seems to have gotten bigger in recent years, do you think that we currently have the answers to the problems in the toolbox, or do we need to see even more new modalities in delivery emerging?
Well, I think we have lived through decades of viral vectors, and that their success is not in doubt. They and if you have a genetic disease or perhaps a cancer that requires continual expression of a Cas protein or a base editor, then those vectors have some life to them because they're going to be consistently produced. Where my own work and our own group started to think about this was that, one of the concerns, certainly at the regulatory agencies, is the so-called off-target mutagenesis. So if you have a constant expression of Cas protein in the CRISPR RNA, then you have a chance of chopping up the chromosome at different sites. Where if you have a non-viral particle with mRNA and CRISPR RNA, it's really a one-time shot. And if you can get the efficiency high enough, then you avoid the secondary type of events. So we waited a long time for non-viral particles. I am old enough to remember the liposome, emerging from Leif Wong's lab at the University of Pittsburgh. And that was an enormous thing. Well, it turned out to be a great reagent to transform cells, but it didn't really transform or transition into human trials. I think the COVID vaccine work actually has transformed gene editing because the use of a non-viral particle, a non-viral vehicle, let's say, to transmit messenger RNA for the vaccines really convinced us. And then watching Intellia and Verve, two companies who have been very successful in targeting liver-based diseases with LNPs, convinced us that the future is probably non-viral vectors. And we certainly have moved to that as well. And we're more interested in localized delivery, whether they're pumps for glioma or head and neck cancer, lung cancer. So I think we're at the precipice. In my world, we watch very carefully these non-viral vectors, that particularly LNPs that are lipids, frankly, that are being developed that target specific tissues. And as you mentioned, it's the arrival at the tissue, but it's also, or the organ, the affected organ, but it's also the penetration of the organ in the tissue. So there are two steps there. I've seen many publications that show we can target specifically lung or heart or, spleen or kidney, but the penetration of the technology into the cells, particularly when you're dealing with a tumor, is as important. So I think that, again, in sort of thinking about, we were talking about universal T cells, I think in the same way, a tangential field that's running alongside a parallel field, if you will, I think in developing LNPs that are able to accumulate, to target, and to penetrate specific tumors or specific organs is on the horizon. And I think that should be a really active way. So we're optimistic about that happening.
Yeah, that's great. I mean, we can see that Verve now have a ligand attached to their newest candidate going in the clinic. So hopefully we will see more ligands for other non-liver organs emerging. I think it will be really interesting to see how it pans out when we see more in vivo candidates entering the clinic, because I think that's really where we're going to need this differentiation when it comes to delivery, because the vast majority of programs in the clinic right now are still the ex vivo cell-based approaches. So just to shift track a little bit, ultimately the goal for any of the many gene editing therapeutic companies out there, like yourselves, is to bring new treatments to the market. How easy is that, though, for a drug developer to do in a field that's still quite in its infancy? And given also the fact that the long-term implications of editing the genome still remain unknown, how, in your opinion, are the regulatory bodies doing enough to guide prospective sponsors in this area?
I have enormous respect for the people who sit on those panels, and we followed very carefully the Vertex application and the discussions there. And with a great deal of admiration and respect, I would say that we're building the plane while we're flying it. So we're kind of learning how to do this, and the regulatory agencies are providing great guidance. I think the desperation of people who have no options, who are, in a case of a lung cancer or head and neck, that have 6 to 12 months to live. What I learned here at Christiana Care and taught me a lot about combining gene editing with patient care was that, sometimes going after, I would say, low-hanging fruit, but advancing survival, progression-free survival for 3 to 6 to 12 months is really the essence of what's going on. But I do think that they're trying really hard, and like you said, you don't know all the cards in your deck. I mean, you're trying to figure out. One thing I think that there are kind of two minor things, but certainly what on a challenging side for our regulatory agencies is that when a new technology emerges, particularly in the DNA sequencing world or an RNA-seq or some sort of analytical tool, there tends to be a rush that everybody has to use that to confirm there's no off-target stuff. And I think you mentioned that whether it's a small company or whether it's a major pharmaceutical company, there's a big difference on who can afford to do that.
Yeah.
So what I'm hoping and what we're beginning to see, certainly at the U.S. FDA that we deal with, is they're tending to look at these protocols a little more as unique protocols. So there isn't a special thing. In our case, we're interested in mid to late stage cancer patients and increasing, and supporting progression-free survival. But a child, and so off-target mutagenesis, for that person may not be as critical as those, as you mentioned before, with a child with sickle cell disease. And
Yeah.
that's specifically why we chose to begin our first work in a patient group that had no option,
Yeah.
but we're at a position in their life that we could improve, maybe in a serious way, but
Mm.
not have to worry about the long-term effects. So I think the change from the regulatory agencies, where they're looking at, this isn't just how you develop, the next aspirin or the next sort of painkiller. It's really about, geez, these guys at Corixa Therapeutics are, they're a small entity. They may have a really exciting idea. It's working pretty well. Maybe the constraints that we put on them advancing that to approval will be slightly different than a bigger, entity that's going after diabetes or something like that. So
Right. Right.
I think that's hopeful, and I think that's, again, because people have learned while they're moving the approval process forward in a brand new and very exciting
Yeah.
area.
Yeah. So really, the prospective sponsor, whoever's developing the therapy, should be engaging with the regulatory authorities early
Abs-
on to find out what
yep.
do you need from us, because there is no one size fits all approach. So we may actually see differential regulation, as we see already in the small molecule drugs for cancer, so
Right.
compassionate base use. I think this has been really interesting. I can see that we are starting to run out of time, but we've obviously could have continued this conversation for at least another few hours. So I do hope you will come back and speak to us again at some point. Perhaps we can hear more about what Corixa is doing at a future CMN live session. I would like to take the opportunity to thank all of you for listening in. This has been really enjoyable, and I hope, for me at least, and I hope that you've all enjoyed it as much as I have. And Eric Kmitch from Christiana Care and also Corixa Therapeutics, thank you so much for taking the time this morning. And I'm very much looking forward to meeting you in person in Copenhagen this April at the CRISPR Med conference. So for any of you who have not already heard about it, CRISPR Medicine News is hosting the world's first in-person conference, bringing together scientists from academia and industry working towards advancing gene editing as a therapeutic modality. And if you hit the QR code on the screen, it will take you straight to the conference website. So with that, I will say thank you once more, and have a good weekend.
Very good, Karen. It's been a great pleasure. Thank you.
Take care. Bye.